In this video, I'll review concepts from Chapter 2 of Human Anatomy and Physiology, 11th edition. This chapter focuses on the chemistry of life. In particular, in this video, we'll be looking at organic compounds that are important to life. I'll remind you that these organic compounds are ones that contain carbon that is covalently bonded to other atoms. So, every organic compound is going to contain carbon. I mentioned in a previous video that exceptions to this are carbon dioxide and carbon monoxide. Note that these two are considered inorganic molecules. One way I like to remember this is that typically in a class like Anatomy and Physiology, you're always going to see both carbon and hydrogen. So at least one carbon, at least one hydrogen in an organic compound. So these two don't fall into that. So carbon dioxide and carbon monoxide are inorganic compounds. Carbon, it turns out, is the perfect element for life. That's because it can bind up to 4 other elements, so it has 4 electrons spinning around it that are able to form covalent bonds with other elements. So those covalent bonds mean that it's sharing electrons. So the carbon never actually gains or loses electrons, it's just sort of sharing them. So it partially pulls them toward itself or partially pushes them toward another molecule. And those covalent bonds, because of their strength, they're not too strong so they can be broken if needed, but they're strong enough that they can form really long chains. So you can get things like a DNA molecule that can be more than 1 meter long. Or it can be very long, as tall as a person, basically, these chains of atoms. And that's thanks to the covalent bonds that are being formed with carbon. So it turns out that it's perfect. It can bind to, each carbon can bind a lot of other elements, and those bonds are just strong enough that they can create long chains while still being broken apart if needed. The organic compounds for life are going to include 4 main categories. So we've got lipids are one category off on its own. So those things include fats and oils, and then macromolecules. And macromolecules is an overarching group that includes carbohydrates, proteins, and nucleic acids. So the 4 groups are carbs, proteins, nucleic acids, and lipids. But as you'll see in a moment, these first 3 are linked by their structures. And they are all part of a group called macromolecules. So what is a macromolecule? One thing, it's not lipids. So lipids are not macromolecules, but the other 3 are. And we define it by being formed, these molecules are formed by 1 or more monomers. So we have these building blocks that can be basically linked together. I like to think of them like, building a structure out of Legos. Each Lego block is 1 monomer. And what you build from those monomers is going to be a polymer. So one thing I will note for you is that each of these monomers may contain many atoms. So there might be, let's say, 6 carbon molecules within this single monomer. But that monomer can now go and bind to a different monomer to form a polymer. So polymers are going to have 2 or more monomers linked together by this covalent bond. So just note this type of definition right now. We have the building blocks, which are like the Lego blocks, the monomers, and the polymers, which are sort of like the Lego structures that are built from the blocks. Those are going to be 2 or more monomers in a chain that is linked by covalent bonds. So this is the definition of macromolecule, and hopefully what you'll see is that this more generic definition is going to apply to carbohydrates, proteins, as well as nucleic acids. So this is how they're all forming their structures. So let's start with carbohydrates. Carbohydrates are important because they are the major fuel sources for your body, and they're also structural molecules. So they are categorized by the number of monomers within the carbohydrate. So if it's just a single monomer, we call that a monosaccharide. That means it has a single sugar monomer. A disaccharide is going to have 2 sugar monomers. So di means 2, so 2 monomers. And then a polysaccharide is going to have many monomers linked together by covalent bonds. So that, in our language, means 3 or more sugar monomers that are linked together. So these are the different names for carbohydrates, and it's, again, based on how many monomers are linked together in a chain. 1 is monosaccharide, 2 is disaccharide, and 3 or more is polysaccharide. Some important examples of carbohydrates, one of the most important is glucose, and that's because it is the major cellular fuel. So it is used for generation of energy in the cell. We also have deoxyribose and ribose. So those are carbohydrates that actually contribute to the structures of DNA and RNA. So you'll see that nucleic acids also contribute to these structures, but those DNA and RNA molecules also rely on these two carbohydrates. So deoxyribose goes with DNA, that's what the D stands for. And RNA is ribose, so R in RNA stands for ribose. Sucrose is another important one, so sucrose is table sugar. And glycogen is a third important, or well, fifth important from this list, carbohydrate. And that's because it is formed by linking a whole bunch of glucose monomers together to form glycogen. So it is the major storage carbohydrate in animals. So if we want to store glucose in our bodies, we're going to do so by forming it into links of, or into chains of glycogen. So here you can see an example down at the bottom where we've got these links of glucose forming the glycogen molecules for storage. Now moving on to proteins. This is the second of 3 types of macromolecules. Proteins are going to have a lot of different possible functions. So proteins can be structural. So you'll hear a little bit about that in a future chapter, thinking about the cytoskeleton of your cell. It's made up of proteins. Additionally, collagen. This is often found in the extracellular space, and it is providing strength in connective tissues. We have enzyme proteins, so these are things that are going to catalyze reactions. We have transport proteins. So I mentioned hemoglobin in a previous video, but hemoglobin is going to transport oxygen around the body. Contractile proteins, so actin and myosin are 2 examples. Together, they're going to form a structure that allows for contraction of your muscles. We have communication proteins, so these are proteins that might bind to a messenger or some sort of molecule on the outside of the cell and deliver that message to the inside of the cell. And we also have defensive proteins, so antibodies are the best known example, where these proteins go and bind to, let's say in this case, a virus, and they alert your body to attack that virus. So lots of functions for proteins. Proteins, so I mentioned carbohydrates, the monomer of a carbohydrate is that monosaccharide. For proteins, the monomers are called amino acids. So here we have a generic amino acid. So you can see it has multiple parts. It has this amine group, which is NH3. We have an acid group, which is COOH. And then there's some sort of side chain. So that's this R group. And that's what's going to make one amino acid different than another. And it turns out that there are 20 different types of amino acids that are central to life. And that means that there are 20 different R groups that you will find in amino acids in your body. The monomers are joined together by covalent bonds that we call peptide bonds. So another way of talking about proteins is to talk about polypeptides. So we have an amino acid is the monomer. Here we have a dipeptide. That means there are 2 monomers in the chain. And then we can also have polypeptides, which would be 3 or more monomers in the chain. All right, and so now we get to the third of 3 types of macromolecule, and that's a nucleic acid. So nucleic acids are responsible for storage, transmission, and expression of genetic information. And there are 2 classes of nucleic acid. We have DNA and RNA. Both of these are built from monomers that are called nucleotides. So you'll notice we've had monosaccharides for carbohydrates, we've had amino acids for proteins, and now nucleotides are the monomer that is used to build nucleic acids. So these nucleic acids, you can differentiate them, so DNA from RNA, one, by location. So you'll find DNA within the nucleus, whereas much RNA is going to be found out in the cytoplasm, although actually it is formed in the nucleus. But much of it will be found in the cytoplasm. Major functions, DNA is going to be the genetic material, so it's the hereditary material. Whereas RNA is sort of a middleman. It's carrying out the directions. It's translating the directions from DNA to protein synthesis. DNA is formed into a double helix, you may have heard of, so that means it has two strands that are coiled around each other, whereas RNA is going to have just a single strand. So not a double helix, but just a single strand of nucleotides. A big difference on the structure would be the sugar involved. So I mentioned this in a previous slide here, but deoxyribose is the sugar found in DNA nucleotides, whereas ribose is the sugar that is found in RNA nucleotides. And then finally, we can see in DNA, we have different bases that are found within these nucleotides. So these bases include adenine, guanine, cytosine, and thymine, so A, G, C, T. If you take a look in RNA, it's AGC, but then uracil, or U, replaces thymine. I'll mention again that DNA is the genetic blueprint for all protein synthesis. Here in this illustration, you can see that double helix. So you can see there are actually two strands of DNA that are coiled around each other in that double helix. It is built from strings of nucleotide monomers. So here's a nucleotide right here. You can see each nucleotide is going to have that central sugar group. So deoxyribose is what's found in DNA. That deoxyribose is bound to a base, in this case it's adenine in the example, and to a phosphate group. So there's the phosphate group on the left side of the nucleotide. So all nucleotides will have those three parts. And you can see that they line up. And when you are looking at this chain, you'll see, once again, I'll mention one of four nitrogen bases. So A, G, C, or T. You'll also be able to see another pattern as you look down this particular example. So whenever you see an A on one strand, you'll always find it paired with a T on the other strand. Similarly, if you find a C on one strand, you'll always find it paired with a G. So A always pairs with T, and G always pairs with C. And in this way, you can ensure that whenever you copy the DNA, that the copies remain as close to the original as possible. So the hope would be, as you copy it, you're always adding the correct complementary base to the strand. So you'll always add a T where you find an A, you'll always add a G wherever you find a C. RNA, as I've mentioned, is going to link the DNA blueprint to protein synthesis. So RNA is just single-stranded, so instead of having this double helix with two strands, RNA will just include one strand of nucleic acid. It's built from, again, the strings of nucleotide monomers, just like in the last slide. The big difference here, you still see phosphate and you still see a sugar. However, the sugar is now a ribose group rather than deoxyribose. Additionally, when you look at the options for nitrogen base, you still see A, G, and C, and G is still going to pair with C always. But now there's a uracil instead of a thymine. And so this means that for mRNA, or for RNA, adenine is now going to pair with uracil whenever it does bind to another nucleotide. So here's just an overarching summary of these macromolecules. So carbohydrates, the monomers were monosaccharides, and the polymers were polysaccharides. Proteins, monomers are amino acids, and the polymers are polypeptides or proteins. And nucleic acids, the monomers are nucleotides, and the polymers are either DNA or RNA. And I'll remind you, once again, lipids are not included on this table because they don't form polymers. So now a question before we move on to lipids. which of the following is not matched correctly? So we have monosaccharide with carbohydrate, amino acid with protein, nucleotide with nucleic acid, and nucleic acid with protein. So one of those is not matched correctly, so select that. And now we'll move on to lipids. So there are three important classes of lipids that have a lot of important functions. First, triglycerides. They're used for storing cellular energy, as well as protecting and insulating body organs. Next up, we have phospholipids. These are the chief component of cell membranes. So most of the cell membrane is made up of phospholipids. And then finally, we have steroids. These are also a component of the cell membrane, and they are participants in cellular and hormonal communication. So focusing on triglycerides. Triglycerides have a particular structure, and they are formed by taking 3 fatty acids, so here 1, 2, 3 fatty acids, and binding them all to one glycerol molecule. So together, these 4 different molecules bound together are called a triglyceride. Triglycerides are called fats when they're solid. So say, butter at room temperature is pretty solid, that would be a fat, versus oils when they are liquids. So olive oil, for instance, would be a triglyceride that is called an oil. The main functions for triglycerides are energy storage, insulation, and protection. Looking in a little more detail, we can say that triglycerides can be either saturated or unsaturated. So for this class, take a look at these 2 pictures and you'll see a big difference. And the big difference is highlighted in the illustrations. So in both cases, they're both triglycerides, so they both have the glycerol plus 1, 2, 3 fatty acids. But for a saturated fat, such as what you would find in butter, you can see that all of these fatty acids are straight chains, so no kinks in the chain. On the other hand, in an unsaturated fat, such as what you find here in this picture, olive oil, we have a double bond that is formed between 2 carbons in this 3rd fatty acid chain. And what that does to the molecule is that it creates a bend. So there's a bend in this molecule. So animal fats, as shown up on top, are mostly saturated. That means they're mainly solid at room temperature, and that's because of this molecular difference. So because all of these fatty acids are straight, that means you can really push them in and push them in tight. So think of like a crowd at a concert. If everyone's holding their arms at their sides, they can really stack people in very closely to each other, shoulder to shoulder. Whereas in this case, you could imagine, let's say someone starts holding their arm out to their side. That would be like an unsaturated fat, where you have a bend in one of the fatty acids. This is going to make it so that these fatty acids can't get so close together. And so this is what you find in many plant fats. They're unsaturated. So they have just extra space between the different triglycerides. And this means that they will be liquid at room temperature. So thinking about health, we used to always say unsaturated fats are good, saturated are bad. Turns out that that was not correct. That was too simplified. There are some saturated fats that are bad. However, there are some saturated fats that are actually healthy for you. So for instance, omega-3 fatty acids, these are unsaturated, and they're found in fish. These actually are now considered pretty healthy for you. On the other hand, we used to say unsaturated fats were all healthy. And so at the grocery store, you would buy margarine based on unsaturated fats because it was a healthy alternative to butter. Well, now we know that's actually not true. Many of those margarines were actually formed from trans fats. So this is a type of unsaturated fat that is considered very unhealthy. And so these are some relationships with health. So we used to have a very simplified view of saturated versus unsaturated, but it turns out it's really not that simple. So next up, we have this second type of lipid, which is a phospholipid. I mentioned before they're the chief component of the cell membrane, so they're vital for cell membrane structure. These phospholipids also have a very distinct structure. So they're always going to have a phosphate group with some sort of nitrogen-containing group. So you can see here, and that's bound to glycerol. And this is going to form what's called a polar head group. So this is a water-loving group. So it's polar, meaning that it's going to try to associate itself with water. On the other hand, we have glycerol bound to 2 fatty acids. So remember, it was 3 fatty acids for a triglyceride. For a phospholipid, just 2 bound to the glycerol. The third space is taken up by this phosphate group bound. But those fatty acid tails are going to make up this non-polar tail. So we have a hydrophilic head and hydrophobic or water-fearing tails. And that's really what defines a phospholipid, is it's got these 2 sides, one water-loving, one water-fearing. And that is perfect for forming a cell membrane. So your cell membranes are going to be formed from a phospholipid bilayer. What that is going to allow, so here's the word bilayer down at the bottom. What that's going to allow is that means outside the cell can be water-based, and inside the cell can be water-based, but it's not going to allow many molecules to pass through this lipid bilayer because there's this hydrophobic water-fearing center to the cell membrane. So this means that whenever a molecule comes up to the membrane, it's going to have a lot of trouble getting through, because if it's out here in the cytoplasm, that molecule probably is hydrophilic because it's interacting with water. So once it gets to this hydrophobic part, it won't be able to pass through. It's going to get pushed back to its own side. So this is going to be a really great setup for dividing the inside of the cell from the outside of the cell, and for really allowing for control of what gets in and out of a cell. Now we have our third and final category of lipid, and that's a steroid. Steroids have a long series of functions. All steroids are going to include 4 interlocking rings. So this is, again, looking at the molecule of a steroid. And in particular, what we've got here is cholesterol, actually. So we have these 4 rings. So these are going to be common in all steroids. Cholesterol, it turns out, again, maybe you could argue this, but it's the most important steroid. And that's because it is the source of all steroid hormones. It's the source of vitamin D, and it's the source of these things called bile salts, which are important for digestion. So that cholesterol, it's not just clogging up your arteries, it's playing a major role in the structure of your cell membranes, and it is the source of all of these other molecules that are vital for life. Now finally, I'm just going to talk a little bit, maybe just this one slide, this one slide about ATP. And this is called adenosine triphosphate. So here's that spelled out. Adenosine triphosphate is ATP. What this means is it has an adenosine group, so that's adenine plus ribose, so adenosine, plus 1, 2, 3 phosphate groups that are all lined up in a chain. These phosphate groups are bound by these high energy phosphate bonds. What that really means is when you break these bonds, a huge amount of energy is going to be released. And our bodies our cells have specialized their enzymes to be able to utilize the energy that's released by these bonds to get their work done. So you can see here, ATP is adenosine triphosphate. So again, 1, 2, 3 phosphate groups. If you break one of those bonds, you're left with adenosine diphosphate, so 2 phosphates groups, so adenosine plus 1, 2, and adenosine monophosphate is that adenosine plus 1 phosphate group. So every time you cut one of these phosphate bonds, that's some energy that can be used for cellular work. So I'll now leave you with a question. Which lipid serves as the source of all steroid hormones and bile salt? And your choices are phospholipid, cholesterol, triglyceride, or glycerol.